There is a particular exhaustion that belongs to people who have been managing eczema for years without ever achieving what could genuinely be called control. Not the exhaustion of a single bad flare, though those are bad enough, the skin raw and weeping, the itch so relentless it disrupts sleep for nights at a stretch, the visible inflammation that draws unwanted attention and requires explanation. The deeper exhaustion is the one that comes from having tried everything that was recommended, having followed every protocol, having eliminated the obvious triggers, having applied the prescribed topical corticosteroids conscientiously, and still finding that the skin never fully settles, that flares arrive without obvious cause, that the baseline level of inflammation never quite reaches the calm that should theoretically be achievable. If that is where you are, there is a biological connection that rarely gets explored in the standard eczema management conversation, not because it is obscure or unproven, but because it sits at the intersection of two medical specialties that do not always communicate well with each other, and because the testing required to identify it is not part of the routine workup that most dermatology appointments have time to include. That connection is between histamine, the inflammatory mediator most people associate exclusively with hay fever and allergic reactions, and the chronic skin inflammation of atopic dermatitis. And understanding it properly requires looking at histamine not as a simple trigger of acute allergic responses, but as a broader mediator of immune activity whose regulation involves the gut, the diet, the microbiome, and a specific enzymatic pathway that varies significantly between individuals. Histamine is a biogenic amine, a small molecule derived from the amino acid histidine through a single enzymatic step performed by an enzyme called histidine decarboxylase. It is produced and stored in several cell types throughout the body, most abundantly in mast cells, which are distributed throughout connective tissue and are particularly concentrated in the skin, the gut lining, the respiratory mucosa, and the brain. Basophils, a type of circulating white blood cell, also store and release histamine. In the skin specifically, mast cells are present in high density in the dermis, the layer of tissue beneath the epidermis, and their activation and degranulation, the process by which they release their stored histamine and other mediators into the surrounding tissue, is one of the central events in the acute inflammatory cascade of atopic dermatitis. When mast cells degranulate in the skin, the released histamine binds to histamine receptors on sensory nerve endings and produces the itch signal through a mechanism involving C-fiber activation that is distinct from the pain pathway but equally compelling in its ability to dominate conscious attention. The same histamine acts on blood vessels to produce the vasodilation and increase permeability that creates the redness, warmth, and swelling characteristic of an eczema flare. And histamine acts on immune cells to modulate the broader inflammatory response in ways that can sustain and amplify the inflammatory state well beyond the initial trigger. The standard medical framework for histamine in the context of eczema focuses on this mast cell degranulation model, the idea that allergen exposure or other triggers activate mast cells in the skin, releasing histamine that drives itch and inflammation, and that antihistamine medications blocking histamine receptors can provide symptomatic relief. This framework is partially correct and clinically useful as far as it goes, but it captures only one dimension of the relationship between histamine and atopic dermatitis. And for people whose eczema does not respond adequately to standard allergen avoidance and topical treatment, it may miss the dimension most relevant to their particular situation. The missing dimension involves the systemic histamine load, the total amount of histamine present in the body from all sources simultaneously, and the mechanisms through which that load is regulated and can become chronically elevated in ways that maintain a pro-inflammatory state in the skin independently of any single external allergen trigger. Histamine enters the body not only through endogenous production by mast cells and basophils, but also through dietary intake because histamine is a natural byproduct of bacterial fermentation and protein degradation that is present in significant quantities in a wide range of commonly consumed foods. The foods with the highest histamine content are those that have undergone fermentation, aging, or prolonged storage, processes that allow bacteria to convert histidine in food proteins to histamine over time. Aged cheeses, particularly hard and semi-hard varieties that have undergone extended maturation, contain among the highest histamine concentrations of any food category. Fermented meats, including salami, pepperoni, and other cured products, accumulate histamine through similar mechanisms. Fermented vegetables including sauerkraut and kimchi, fermented soy products including miso and tempeh, fermented beverages including wine, beer, and kombucha, and fish products, particularly those that have been smoked, canned, or stored for extended periods, all deliver meaningful histamine loads with consumption. Certain foods including tomatoes, spinach, eggplant, avocado, and strawberries contain histamine directly or contain other biogenic amines including tyramine and putrescine that share degradation pathways with histamine and can compete for its enzymatic breakdown, effectively increasing the net histamine burden even when their own histamine content is relatively modest. The critical variable determining how much of this dietary histamine reaches systemic circulation and contributes to total body histamine load is the activity of an enzyme called diamine oxidase, abbreviated DAO, which is produced by the enterocytes, the absorptive cells lining the small intestinal mucosa, and secreted into both the intestinal lumen and the bloodstream. DAO is the primary enzyme responsible for degrading histamine in the intestinal environment, oxidatively deaminating it to imidazole acetaldehyde and preventing its absorption into the portal circulation. When DAO activity is adequate, the enzyme breaks down dietary histamine in the gut lumen and at the intestinal surface before significant quantities can be absorbed. When DAO activity is insufficient, whether because of reduced enzyme production, competition from other substrates, or inhibition by certain compounds, dietary histamine is absorbed into the portal circulation, reaches the liver and systemic circulation, distributes to target tissues including the skin, and adds to the endogenously produced histamine pool in ways that can maintain a chronically elevated histamine burden without any single dramatic exposure producing an obvious acute reaction. DAO deficiency, or more precisely the state of inadequate DAO activity relative to histamine intake, is the central concept in what clinicians and researchers have called histamine intolerance, a condition characterized by a reduced capacity to break down dietary histamine efficiently, producing a wide range of symptoms that reflect histamine's actions on multiple organ systems simultaneously. The symptom profile of histamine intolerance is deliberately broad because histamine receptors are distributed throughout the body and histamine has different effects in different tissues. Skin manifestations including flushing, urticaria, and exacerbation of existing inflammatory skin conditions including eczema are among the most common. Gastrointestinal symptoms including abdominal cramping, bloating, diarrhea, and nausea reflect histamine's effects on intestinal smooth muscle and secretory function. Headaches and migraines are among the most frequently reported symptoms, driven by histamine's vasodilatory effects on cerebral blood vessels. Cardiovascular effects, including palpitations, rapid heart rate, and blood pressure changes, reflect histamine's actions on cardiac and vascular tissue. Rhinitis and nasal congestion reflect histamine's effects on respiratory mucosa. This multi-system symptom pattern, variable in its expression between individuals and in its severity depending on the cumulative histamine load on any given day, is part of why histamine intolerance is frequently not recognized as a unified condition. The symptoms are attributed to multiple different diagnoses rather than to a single underlying mechanism of impaired histamine degradation. The relevance to eczema specifically is supported by several lines of evidence. Studies measuring DAO enzyme activity in people with atopic dermatitis have found significantly lower activity compared to people without the condition, suggesting that impaired histamine degradation capacity may be a feature of the atopic disease state rather than simply a coincidental finding. The gut microbiome, whose disruption is increasingly recognized as a feature of atopic dermatitis, children and adults with eczema consistently show reduced gut microbial diversity and altered microbiome composition compared to controls, influences DAO production and activity through mechanisms that are not yet fully characterized but likely involve bacterial metabolites affecting enterocyte function and intestinal permeability. Increased intestinal permeability, which allows larger molecules including incompletely degraded food proteins and bacterial products to cross the intestinal barrier and enter the systemic circulation, is documented in atopic dermatitis and creates a route through which histamine and histamine-liberating compounds can reach the immune cells in the skin that drive inflammatory responses. The skin barrier defect characteristic of atopic dermatitis, driven largely by mutations or reduced expression of the structural protein filaggrin, allows external histamine from the environment and histamine released by activated local mast cells to reach sensory nerve endings and immune cells more easily than in intact skin, amplifying the effect of any given histamine exposure. The atopic individual therefore often faces both a higher systemic histamine load due to impaired degradation and a more histamine-sensitive target tissue due to the barrier and sensory abnormalities of eczematous skin. The second major enzyme involved in histamine degradation, histamine N-methyltransferase, abbreviated HNMT, operates primarily intracellularly rather than in the gut lumen and is responsible for histamine breakdown within tissues, including the skin itself. HNMT transfers a methyl group from S-adenosylmethionine to histamine, producing the inactive metabolite N-methylhistamine. Genetic variants in the HNMT gene that reduce enzyme activity have been identified and studied in relation to atopic dermatitis risk, with some variants associated with increased disease severity, providing a genetic basis for individual differences in histamine sensitivity in the skin that operates independently of dietary histamine load and DAO activity. The practical implication is that histamine management in eczema involves both reducing the amount of histamine entering the system through dietary modification and gut health support, and optimizing the enzymatic capacity to break it down both in the gut and within skin tissue. Several factors are known to suppress DAO enzyme activity and therefore worsen histamine intolerance in susceptible individuals, and identifying and addressing these factors is often more practically impactful than dietary histamine restriction alone. Alcohol is one of the most potent DAO inhibitors known. It both competes with histamine for DAO-mediated degradation and directly suppresses DAO activity, explaining why wine and beer in particular, which combine direct alcohol-mediated DAO inhibition with high intrinsic histamine content, so reliably worsen histamine-related symptoms in sensitive individuals. Certain medications inhibit DAO activity as a pharmacological side effect, including some commonly used drugs whose connection to worsening eczema is rarely recognized. Non-steroidal anti-inflammatory drugs including ibuprofen and aspirin, certain antidepressants, certain antihypertensive medications, and some anti-malarial agents have documented DAO inhibitory properties. If you are taking any of these medications regularly and have poorly controlled eczema, the possibility that they are contributing to histamine accumulation through DAO inhibition is worth raising specifically with your physician rather than assuming the connection has already been considered. Gastrointestinal inflammation from any cause, including the mucosal damage of inflammatory bowel disease, celiac disease, or the intestinal permeability associated with dysbiosis, reduces DAO production by damaging the enterocytes responsible for its synthesis, creating a bidirectional relationship in which gut inflammation worsens histamine degradation capacity and elevated histamine further drives intestinal inflammatory signaling. Nutrient deficiencies relevant to DAO production and activity deserve specific mention because they are common, measurable, and correctable. DAO is a copper-containing enzyme, and copper deficiency reduces its catalytic activity. Vitamin B6, in its active form pyridoxal 5-phosphate, is required as a cofactor for histamine metabolism, and B6 inadequacy impairs the histamine degradation pathway. Vitamin C has been shown to support DAO activity and independently promotes histamine breakdown through alternative pathways, with some research demonstrating that vitamin C supplementation reduces plasma histamine concentrations. Zinc, as discussed in other contexts, supports immune regulation in ways that influence mast cell activation thresholds and histamine release. These are not exotic or expensive nutrients. They are obtainable through a well-constructed diet or straightforward supplementation. But their relevance to histamine metabolism means that nutritional assessment in someone with poorly controlled eczema and suspected histamine intolerance should include evaluation of these specific micronutrients rather than simply general nutritional adequacy. The low histamine dietary approach used clinically in the assessment and management of histamine intolerance involves reducing intake of the highest histamine food categories for a defined trial period, typically 4 to 6 weeks, while simultaneously reducing intake of histamine liberators and DAO inhibitors, then systematically reintroducing food categories to identify individual tolerance thresholds. The foods most consistently recommended for elimination in the initial phase include aged cheeses, fermented meats and vegetables, wine and beer, vinegar and vinegar containing condiments, canned and smoked fish, tomatoes and tomato products, spinach in large quantities, eggplant, avocado, and strawberries. Fresh unprocessed proteins, fresh meat, fresh fish consumed immediately after purchase, eggs, are generally well tolerated. Fresh vegetables, with the exception of those listed above, are generally low in histamine. Most fresh fruits, with the exception of citrus, strawberries, and certain tropical fruits, are tolerable. This is not a permanent restrictive diet. It is a diagnostic and therapeutic trial that establishes whether histamine load is a meaningful driver of the individual's inflammatory burden, and tolerance thresholds vary considerably between individuals such that some people can reintroduce most foods after a period of gut healing and histamine load reduction, while others identify specific categories that consistently worsen their symptoms and benefit from longer-term avoidance of those specific items. The gut microbiome connection to histamine in eczema operates in both directions and is worth understanding bidirectionally rather than as a simple linear relationship. Certain bacterial species resident in the gut produce histamine from dietary histidine. Lactobacillus reuteri, for example, which is otherwise considered a beneficial probiotic organism, expresses histidine decarboxylase and produces histamine as a metabolic byproduct. This does not mean that probiotics broadly worsen histamine intolerance, but it does mean that the specific bacterial strains selected for probiotic supplementation matter in this context, and that probiotic recommendations made without consideration of histamine production capacity may worsen rather than improve the histamine burden in sensitive individuals. Lactobacillus rhamnosus and Bifidobacterium species do not produce histamine and have demonstrated benefit in atopic dermatitis research through mechanisms including gut barrier strengthening and immune modulation. The distinction is specific enough that generic probiotic supplementation is less appropriate than targeted selection of strains with documented relevance to atopic disease and confirmed absence of histamine-producing capacity. A clinician with expertise in the microbiome eczema connection, typically a gastroenterologist with interest in atopic conditions or an allergist immunologist, is the appropriate guide for this level of specificity. Mast cell stabilization represents a complementary pharmacological and nutritional strategy that targets the histamine release side of the equation rather than the degradation side. Mast cells in eczematous skin are present in elevated numbers, are more easily activated, and degranulate more readily in response to stimuli that would not trigger significant histamine release from mast cells in non-atopic skin. Quercetin, a flavonoid present in onions, capers, apples, and several other plant foods, has demonstrated mast cell stabilizing properties in research. It inhibits IgE-mediated mast cell degranulation and reduces the release of histamine and other inflammatory mediators from activated mast cells through mechanisms involving the suppression of intracellular calcium signaling. The research base for quercetin is primarily in cell culture and animal models with some supportive human data, and the bioavailability of quercetin from dietary sources is variable and influenced by the food matrix it is consumed in, with onions providing more bioavailable quercetin than most other sources. Vitamin C at supplemental doses in the range of 1 to 2 grams per day has demonstrated histamine-reducing effects in clinical research, attributed to both direct chemical interaction between ascorbic acid and histamine and support of the enzymatic degradation pathway. These nutritional approaches to mast cell stabilization and histamine reduction complement dietary histamine restriction rather than substituting for it, addressing both the production and the degradation sides of the histamine balance simultaneously. The clinical pathway for someone with poorly controlled eczema who wants to investigate the histamine connection begins with a conversation specifically framed around histamine intolerance and DAO activity with either their dermatologist, their allergist immunologist, or a gastroenterologist with interest in food intolerance. DAO enzyme activity can be measured in serum, and while the test is not universally available and its clinical interpretation requires some nuance, it provides objective information about enzymatic capacity that is more reliable than symptom history alone in establishing whether impaired histamine degradation is genuinely contributing to the clinical picture. Plasma histamine levels can also be measured, though they are highly variable and must be interpreted alongside DAO activity rather than as an isolated value. A comprehensive food allergy and intolerance evaluation, including both IgE-mediated allergy testing and consideration of non-IgE-mediated food reactions, provides the broader context within which the histamine intolerance question sits. Because histamine intolerance frequently coexists with IgE-mediated food allergy and with other non-allergic food sensitivities that contribute to the overall inflammatory burden in the skin. The clinical reality of atopic dermatitis management is that it is almost always multifactorial, driven by some combination of genetic barrier dysfunction, immune dysregulation, microbial dysbiosis on the skin and in the gut, allergen sensitization, environmental trigger exposure, and in some individuals, the dietary and metabolic histamine burden that this discussion has focused on. No single intervention addresses all of these dimensions simultaneously, which is why the most effective management approaches are comprehensive rather than sequential, addressing multiple contributing factors in parallel rather than trying each one individually. The histamine connection is one dimension of that comprehensive picture that is frequently underexplored and sometimes highly relevant. Not universally the missing piece for every person with eczema, but specifically and meaningfully relevant for those whose disease has not responded adequately to standard approaches and whose symptom pattern and dietary history suggest that histamine load may be a significant contributor to their ongoing inflammation. For that person, the low histamine dietary trial, DAO activity testing, targeted microbiome support with histamine-neutral probiotic strains, nutrient repletion of the cofactors supporting histamine degradation, and review of medications with DAO inhibitory properties collectively represent a coherent and evidence-informed investigative and therapeutic approach that can be pursued alongside, rather than instead of dermatological care. The skin is not an island. It reflects the inflammatory state of the whole body, and the histamine connection is one of the most direct pathways through which what happens in the gut and in the diet expresses itself on the surface of the skin. Understanding that connection clearly is the beginning of being able to address it effectively. If this opened a dimension of your eczema management that had not previously been explored, share it with someone else navigating the same cycle of inadequate control and unanswered questions. The conversation with your physician that follows from understanding this biology is a more specific and productive one than the conversation that preceded it, and that specificity is where better outcomes begin.